Good morning. Let us continue with the histology series. Last week we talked about the histology of the small intestine that consisted of the duodenum, jejunum and ileum. We said the pathognomonic feature or the characteristic feature of this part of the gut is villi, the presence of villi, which is surface folds of mucosa which vastly increase the absorptive surface or the interactive surface between the contents of the lumen and the surface lining of the gut. Now this 5 meters of tract is a slow gradation of histology from its duodenal or pyloric end to its ileocecal junction end. After the ileum is over, we come to the next part of the intestine, which is called intestine, which consists of another series of organs in sequence, one behind the other, where the processing of the contents of the gut continues and as much of material from it as possible is absorbed, and then it is lubricated to exit as a semi-solid substance from the anal canal or the anal orifice. So what are the parts of the large intestine? Since we haven't done it in gross anatomy, I will just go over it for a few minutes before we launch into its microstructure. So I think you already are familiar with it. We have a proximal portion called the cecum just after the ileum. Then we have an ascending colon, a transverse colon, a descending colon, point colon, and a straight part called the rectum, which ends in the anus. It's about 5 feet in length, this whole thing, which is about 1.5 meters. First segment is the cecum, after which there is an appendage, a small finger-like process attached to the cecum, which is probably we can say one of the foremost or the proximal most part of the large intestine. Then we have the colon, which is consisting of these 3 segments. These are named as per how they are arranged inside your abdominal cavity, but they have a continuum of structure in terms of their wall, the 4 layers, and the internal mucosa are entirely exactly the same. There is no change. Then we have the rectum, where there is a beginning of a change, and then you have the anus or anal canal, where because it is opening to the outside, there will be a transition to a surface kind of later. Now, as far as our histology classes are concerned, we'll talk about the structures, but in your practicals, we will be showing you slides of the colon and of the appendix, because there is a marked or a significant difference between the two, largely because their function also differs. So what we see is that the function of the large intestine is basically absorption of water, formation of the feces, and production of mucin, which is a protein, a slippery, slimy protein, facilitates the defecation of the semi-solid fecal mass. So this shows also the blood supply of the gut. It's the gut taken out and pinned in its entirety to show you the whole length and what it looks like with more or less natural life colors. But as we always say, the function is what subserves the structure. So we will see that the gross anatomy, the large intestine has 3 main morphological features. If you look at the outside of it, you see that there are bands of vertical muscular structure. It looks like strips of muscular tissue. They are called the tinea coli. Then we have the horstria. We have these pocket-like distensions, saculations of segments of the gut between the outpocketings will be seen between the tinea. There are 3 vertical tinea, and in between the tinea, the wall of the large intestine will bulge out in the form of sac-like pouches, which are given a name called horstria or saculation. And we see some fat-filled attachments on the surface of the outer wall, which are called the appendices epiploicae. Nothing but there would be a mesentery, there would be a peritoneal fold covering the gut. It gets duplicated in certain areas, and between those 2 layers of peritoneum, a lot of fatty tissue is deposited, and they are called the appendices epiploicae. Now, these are gross anatomy terminology, not to be used in histology at all. I'm just introducing them here so that you are familiar with what the big gut, the large intestine, looks like from the outside. If you look at it from the inside, you see mucosal folds, several mucosal folds, again in a horizontal plane or in the transverse axis of the longitudinal gut, similar to but not the same as the valves of Kerckring which we saw in the small intestine. There, compared to the lumen, the circumference of the small intestine, those folds, mucosal folds, were very big. That means if a fold went into the lumen and came back, it would cross at least 1/3 or 1/4 of the radius of the small intestine. Whereas in the large intestine, these mucosal folds are much less compared to the width of the intestine, but they increase the surface area available. So we have mucosal folds both in small and large intestine, much less in the large intestine without any particular depth. Coming back to microstructure, as we have seen right from esophagus onwards, we must have a 4-layered intestine. We must have a mucosa, then a submucosal layer, then a muscularis externa, and a serosal layer. In that, what we see that the mucosa from the surface of the lumen right till here is having a simple columnar epithelium which dips into the lamina propria in the form of intestinal glands, openings of which appear like pits on the surface. Openings of the intestinal glands, you look at it from the surface of the lumen, looks like small pits. This is similar to how it was in the intestine and even in the stomach, right? There we called it gastric pits. Small intestine we call them crypts or paper. Here we don't give them any particular name. They're just openings of intestinal glands. There is no surface projection called villus. It's extremely important and the main differentiation between small and large intestine. There are no surface projections, but you get this glandular, parallel, simple tubular glandular throughout the length of the whole large intestine, consisting of simple columnar epithelium and a whole bunch of goblet cells embedded in it. In the lamina propria, in between these glands, we will see that there will be the supplies coming in. A little bit of lymphatic capillaries. There will be arterial and venous ends of the capillary. The network where the arterial blood changes to the venous blood. I see them as cut sections or longitudinal sections inside or in between the glands, but not at all as prominent as we saw them in the villi or the small intestine. And we may see lymphatic tissue, lymphocytes, scattered lymphocytes in the lamina propria, as well as aggregated lymphocytes into what are called lymphoid follicles. They will also be seen increasing more as we proceed down the gut. The maximum concentration, however, are seen in only that one organ called the appendix, the vermiform appendix. We will look at that in the slide. But remember that there, the lymphoid available is so dominant that becomes the way we identify. So we will look at that. But throughout the large intestine, we will have lymphatic in the form of nodules as well as in the form of loose lymphocytes scattered in the lamina propria. Then we have the muscularis mucosa, which was the next layer. This is circular, this is longitudinal, 2 layers of muscularis mucosa. But you see, it doesn't seem to be extending in between the glands into lamina propria. Small intestine, we saw that muscularis mucosa was getting into the heart, the core of each villus, and that is an important differentiation. Of course, smooth muscle. Then you have lymphatic vessels, arterioles, venules, some of which you will see, or you will see cut sections of these vessels inside the submucosa. This is the submucosa layer inside which you will see the cut sections of these vessels. You will see them long like this. This is dichromatic. You will see them like cut sections. Then we will see the muscularis externa, again inner circular, outer longitudinal. Please remember this. It's extremely important to diagnose this as a piece of gut, inner circular, outer longitudinal. And in between, we must remember there is a myenteric plexus of nerves. Here there should have been a submucous plexus of nerves. So 2 plexuses of autonomic which supply the smooth muscle gut and which allow it to have changes in its mucosal dimension or diameter, as well as changes in the length of the segments when peristalsis happens. So the longitudinal muscle will squeeze the contents forward from proximal. Circular muscle will squeeze the contents amongst themselves to allow more amount of to happen. So the longitudinal layer, circular layer and the longitudinal layer together forming the muscularis externa, outside of which we expect a serosa or a adventitia layer, that one simple. Much of the large intestine is actually retroperitoneal. That means this the ascending descending colons are lying down on the posterior abdominal wall. So the surface on which they are lying down does not need a mesentery, and it is flush, it is adventitious tissue, connective tissue lying on the fascia of the posterior. But its anterior surface will be covered with a parietal layer of film. So if you look at a segment of the gland, depending on which side of it you may have a serosa or it may not have it. We will look at that in the histology, and we will try to see which part of the gut we have gotten a piece of. So from the luminal side to the external side, this is the progression of the wall of the gut, following the same architecture as we have already seen in most of the previous portions. So if you see a transverse section of the colon at a low magnitude, you will see that there is a mucosa, submucosa, and muscularis, and a very thin serosa or adventitia on the outside. The mucosa is staining bright, and you're seeing these intestinal glands. Right? They're dipping in, dipping out, and they are lined by two types of cells, but you can see a lot of empty looking spaces which are all goblet cells. So all the empty looking gaps are occupied by goblet cells. In low power, that is the size of the cell, each one. 1, 2, 3, 4, 5, 6. So they are narrowly seen in this magnification, but you see that the mucosa as a whole stains well compared to the other layers. Then the submucosa is very sparse because we know it is loose connective tissue, and we should be able to see some blood vessels, cut sections, some adipose tissue. Very clear. This is another vessel. This is another vessel. Some gaps which you see, this may be an artifact, a tear. But these two are blood vessels because you're seeing some tunica media. It's probably an arteriole lying within the circumference. Supplies, arteriovenous, nervous, lymphatic supplies are found in this domain or this slab of tissue. After which starts the muscularis. So muscularis externa is from here to here. Inside we expect circular muscle, outside we expect longitudinal. Now the new thing we are seeing here is suddenly the longitudinal muscle is very thick here, but it's hardly anything over here. Now this is because of what we saw, Tinea coli. It's available in 3 longitudinal sticks. It is a continuous layer around the circumference, but very minimal in between tinea. In the tinea, the longitudinal muscle is most pronounced, and you will see TS of it when you're looking at it. TS of low magnification, you will see here TS of it because the longitudinal muscle is like this, and you cut it like that. So you see the TS of smooth muscle. And in this for circular muscle, you'll see LS of smooth muscle. In this particular slide, the hematoxylin hasn't stained very well, so you're not seeing the purple nuclei. Otherwise, you'll see dash-shaped nuclei in this place and dot-shaped nuclei in this place, and the nuclei will be in the center of this myocyte, in the center of the cytoplasm, not in between the cells like in the skeletal muscle sensation. Smooth muscle in 2 layers. Inner circular is continuous and almost the same thickness throughout, whereas outer longitudinal is interrupted. There is thin layer throughout, but a thick layer in 3 places at the circumference. So this is the labeling: mucosa, submucosa, blood vessels, muscularis externa, consisting of circular and longitudinal. And TC is actually a name of the gross anatomy, morphological structure called Tinea coli. But in this section, we are able to see one tinea in its low magnification microstructure, showing only longitudinal cut and teeth. If we were to see it in a higher magnification, we won't be able to see all 4 parts of the wall together. So we will concentrate on the mucosa as usual. And on the mucosa, we'll see there are no surface projections. There's a kind of a smoothish surface, but it is dipping into the LP as a series of parallel simple tubular glands composed mostly of large number of goblet cells towards the surface and more number of simple columnar cells towards the base. So this is the base of the gland, and this is the apex of the gland or the luminal side of the gland, showing more amount and larger goblet cells. The lamina propria is in between the gland. Yeah, lamina propria, you can see a lot of purple dots. These are the lymphocytes which are scattered. The presence of scattered lymphocytes is one important, it's not diagnostic, but it is an important point in saying that this is colon rather than any other part of the gut. But the shape, the parallelness and the simplicity of these tubular glands is one way of recognizing the large intestine. The mucosa is from there till there and includes the muscularis mucosa, Then we have the submucosa from here to here. One important component is adipose tissue. You get a lot of adipocytes available inside the submucosa, and they increase as you go towards the end. More amount of adipose tissue is available. From here to here is this is the lumen. Okay, in this diagram, this is all we can. We are not seeing any lymphoid follicles. We can see it in another magnification. To understand what is going on, please ignore this, the panel cells we saw in small intestine. But this is just to show you that there is a progression and a replacement of this simple columnar epithelium as it was in small intestine. There is a continuous turnover at the luminal surface because it is abrading with the contents of the lumen. The contents of the large intestine are getting drier and drier. Their moisture, the water content is being reabsorbed. Function, we said, is reabsorption of water. Good amount of water is reabsorbed. You remember, water is one of the 6 classes of your food. So not apart from all the water that you drink, some of it is getting absorbed earlier, higher up in the gut, but the majority of it is being absorbed in the large intestine, and it forms a good amount of the water content of your 75% water content of the body. So that water is going in through your columnar cells, and the goblet cells are not absorptive, but they are secretory in nature. So the two functions being observed here, you have the simple columnar epithelium doing the absorption of water, and the goblet cells doing the secretion of water. Therefore, as the content of the colon proceeds down it, we start calling it fecal mass. That starts getting dehydrated, gets harder and harder, and this mucin coating allows it to be smoothly propelled down the gut right to its end. So we must remember that from the bottom of the gland these cells are being replaced, so that means there must be some replacement cells which are undergoing high mitosis, which can differentiate into goblet or into simple columnar. They move up. Right, so a lot of mitosis will be happening at this level. These pluripotent cells are called stem cells. Please ignore panel cells, they do not occur in the large intestine. So the mucosa, as I said, has 2 types of cells, and as the feces passes along, it becomes progressively dehydrated because of the absorptive cells absorbing water. One more function of the intestine not mentioned in my slide presentation is production of vitamins 12, isn't it? And vitamin K contributes to the formation of 2 important vitamins, but it's not these cells which are doing it. What is doing it is colonic bacteria. We have a whole lot of bacteria within our bodies which are called commensals, which are not harmful to us, which live in symbiosis and which supply some of our nutritive material, but which create vitamin K and vitamin B12 and leave it there in the lumen. That also gets absorbed by our absorptive cells. So that's also a very important component or a function of the large intestine, but not done by our surface cells, done by our guests, those which are living with us but are not part of our biology. Alright. Now the absorptive cells is a very, very high magnification electron microscope to show you that inside the cell there are so many things. The Golgi apparatus, there are lysosomes, yes, brain, a lot of stuff is going on. So this is what an absorptive cell looks like, and is showing you some microvilli kind of processes on the surface which are useful in the absorption. When you see a stained picture, you see that the absorptive cells are tall columnar from here to here, with the nucleus being pushed more to the base. Surface may or may not be able to make out. There may or may not be any surface in this stain. But the other cells are Golgi. Now, this must not be HE, because in HE, goblet cells do not stain at all. So we must have used some special stain to stain goblet cells for this serum. This is the lumen. This is the gland. So this one side, that one side, and this is the lumen. There's some material here. This is not the part of the histology of the gut, right? This is the content of the lumen. This was only to show you what absorptive cells look like. Goblet cells look like this, right? and the nucleus is actually pushed to the base and you're not even able to see it. There'll be a nucleus somewhere here. But this material is the mucin in the supranuclear compartment of each of the goblets. Okay, now there is no content, but whatever they secrete becomes mucin, right? And it gets extruded out into the lumen to mix with the contents which are passing through the gut. So this is what I don't know if you can see the surface at this magnetic, the surface projections from the absorptive. So this is an absorptive cell. This is the solid cell. Now let's come to appendix. This is again a cut section, a transfer section through the entire appendix. So you're seeing this whole lumen because it's only about finger thick. It is not as wide as the large intestine. So if you take a cut section, you see that it is suspended by a two-layered mesentery from the ileum, from the termination of the ileum, because it is available at the ileocecal junction. So the suspension of the appendix makes it covered by mesentery all around and therefore gives it an external-most. Now this small blind-ended tubular sac extending from the cecum just distance to the ileocecal junction is variable in length and in position where it is within the abdominal cavity. If it be 2 inches, it will be 12 centimeters or 12 centimeters. It is quite variable. Your appendix and my appendix may be very different. But their location on the intestine is standard. It is always this. Its function actually till today is there is some dispute, there is a lot of literature, there is a lot of publications. We presume that because of the presence of all these lymphoid follicles, very dominant feature, the lumen is collapsed because of the projection of the mucosa in. When anything enters it, then only it distends and becomes a circular or oval lumen. But immediately under the mucosa, we see purple staining lymphoid follicles. Can you they are ovoid structures. They are lymphoid follicles. Now you've seen them in lymphoid organs long time ago in general history. We saw them as aggregated lymphoid follicles or Peyer's patches in ileum last week. And in the appendix of the colon, see them very dominant. They are throughout the circumference and they are right adjacent to the mucosal. In some mammals, it is supposed to help in digestion of cellulose, etc., which we don't, and humans cannot digest it. So, why we have the appendix seems to be probably of an immune defense thing. In fact, some books describe it as the abdominal tonsil. But this function of it is actually limited only to a short part of your lifespan. These are not visible at birth. If you see your appendix at birth, these things are not visible at all much. And you may see some scattered lymphocytes. It's very clean looking. This only thing is its diameter is much lesser than the large intestine. In development of the colon, it will be explained to you why the appendix is so narrow compared to the rest of the large intestine of which it is a part. But right now you just remember that we recognize it as a separate part in terms of its external dimension. And know that it doesn't seem to do what it does in other animals, but it may be having a defense nature because of the presence of. After the age of about 10 years of maybe teenage, post-puberty, this lymphoid nature also keeps decreasing till the end of your life. So the peak lymphatic or defense activity happening in the pre-pubertal age and extending maybe up to early adulthood, after which the function of defense, secondary defense, seems to decline over the rest of your lifetime. Now the suspensory mesentery is in continuity with the outer serosal layer. There was a serosal layer. Now why it has stained red here is some bleed must have happened, some vessel must have torn, albuces have spread, have extravasated into the layer between the serosa and the muscularis. This is not a normal feature. Please don't think of it as a bloody end. Serosa is actually just looking like that. Simple. Can you see this is a thin line, the simple squamous epithelial line, because that is what constitutes the peritoneum, and it secretes fluid outward into the peritoneum. So that is the outer serosal. The function of these mesenteries is to bring in all the supplies to these suspended parts of the gut. So they're hanging inside a fluid-filled cavity. They need to stay alive. This double sheet of mesentery, anywhere, wherever it is present within the intestine or stomach or large intestine, brings in the vessels, lymphatics and nerves to and from the gastrointestinal tract. So if there's any absorbed food, that will go away in the veins via the mesentery towards the main portal. In this case, it will only absorb water, if at all. But there's not much, much of, in fact, almost never does food actually enter into the lumen of the appendix, because it comes from small intestine into cecum and then goes up the ascending colon. But if at all some does, if some material of food does enter into, it is immediately exposed to the lymphoid follicles which are available here, which can take care of any infectious material or foreign proteins which are coming in. At a slightly higher magnification, we can label the layers. We know that from here to here, from here to here is submucosa, and these lymphatic nodules are occupying the submucosa but spilling into or continuing into mucosa and right up to the surface. Right? All these are lymphocytes. They're coming right. So mainly their location is in the submucosa. Presence is in the submucosa. But they sometimes push through, they interrupt the intestinal glands and come right up to the surface. And this is important because then the surface cell becomes a presenting cell, an M cell. I mentioned it in the last class. They are these macrophage type cells which are surface cells. Actually, they are microvilli cells. You can't call them macrovilli, they're modified. Those microvilli present any antigens or foreign proteins inside the lumen to the lymphoid follicle. So the surface here is simple columnar epithelium and goblet cells, but the surface here will have a lot of M cells because the lymphocytes are present just beneath the chest and they can take care of any foreign protein recognized. So the presence of a mucosa and a submucosa. Otherwise, in the submucosa, you'll see cut sections of blood vessels. So to see some adipose tissue, can't make out. This kind of a big gap is an artifact. This layer of submucosa has been sheared off from the muscle in processing this tissue. Same thing here. This much of a gap doesn't exist. It is more of an artifact. The muscle layer will be immediately deep to the submucosa, and it will consist of inner circular and outer longitudinal. But please see that in the appendix, the outer longitudinal is continuous. There are no tinea coli. There are no hostrations. Okay? And we had used the word appendices epiploicae. That was for the fat-filled folds of mesentery on the outer surface of the large intestine. That is completely different from vermiform appendix. The whole organ is called the appendix. It's an appendage to the whole large intestine. Whereas the appendices epiploicae are small appendages only to the outer wall of the large intestine. Okay? That is a gross anatomy term. This is the name of the full organ. And we must remember that it does not have the three longitudinal muscle bands. It has a continuous external muscularis externa longitudinal layer. Right. The characteristic feature is the masses of lymphoid tissue which form follicles often containing germinal centers. If you see any one of them, you can see the outside is dark purple. That means condensation, the concentration of lymphocytes is dense on the outside, and that is why the inside looks lighter color, stains lighter, and they are called the germinal centers, where the lymphocytes are dividing rapidly to form clonal copies of themselves when required to build up a defense reaction. So you have the germinal centers and the peripheral, let's say the cortex, the germinal center and the cortex of a lymphoid follicle. But that's not right. We only use that word in MIMS. We don't use that word here. Just say germinal center within the lymphoid follicle. That's it. This one doesn't seem to be showing it. It didn't come in the slides. But lymphoid follicle is the main. Large lymphoid follicles compared to any other feature. If you show it at very high magnification, this whole thing is a lymphoid follicle with a lighter and thicker terminal center, a cap of only densely packaged, and it's coming right up to the surface itself, interrupting or separating the intestinal glands from each other. This is the surface gland, exactly how it is in the rest of the colon. So the base architecture is exactly like colon. There will be a mucosa, submucosa. In the mucosa, there will be parallel uh, uh, simple tubular intestinal glands going into the lamina propria. Lamina propria has also a lot of lymphocytes, and it has these sections, cut sections visible of intestine, which also have a lot of goblet cells. That part is exactly like the large intestine. But the presence of these huge lymphoid follicles, much bigger, much more dominant, and coming up to the characteristic appendix part. Here, I showed you this just to show you that the lymphoid follicle may not be very clear in many places, but the lymphocytes are always present in the lamina propria. These are the intestinal glands. It seems to be a tangential section, not a perfect TS. So you're not getting the length of the gland like that. You're getting a bit, and then this part didn't get cut, and then you're getting the bottom of the gland. Okay, what you remember is the structure of the gland. There's only two types of cells. It also has cells undergoing mitosis. It also has a few argent-tapping cells within it. Because we have some neurohormonal molecules like vasoactive inactive polypeptide. Like certain molecules are produced, which we will learn in detail in physiology and biochemistry. But just remember that from the histological perspective, the dominant cell is the simple columnar absorptive cell and the goblet cell, which is producing. Here in this slide, you can see the laminar muscularis separating the lamina propria from submucosa. Otherwise, this domain and this domain don't seem to have any differentiation from each other if you don't see this layer of muscle. So mucosa stops here, submucosa starts here, stops here, where the muscle starts, the circular and then longitudinal. In the submucosa, you will see blood vessels, random connections. There's no adipocyte particular, right? But again, towards the mucosal side, you see a whole lot of lymphocytes. The lymphoid follicles occupy the more luminal part of the submucosa, basal part of the submucosa, the outer part towards the muscle. We'll have more of the arterioles, venules, nerves, and mucosal glands are much less packed than they were in the colon. Okay, this is sideways, but again to show you that at least it's showing you the typical intestinal gland pattern, very typical of colon. These intestinal glands, simple glands, tubular, parallel to each and having only two varieties of cells. Here you have a lymphoid nodule and it is coming right at the surface. Now again, you can use the word cap. Some books give it, but I prefer you don't use the word capsule because that confuses you with capsule organ. You don't even go there. Just talk about it as a lymphoid follicle with a germinal center. The light part is the germinal center. From here to out is the lymphoid. Actually, uh, some books will call it nodule, some books will call it follicle. It doesn't matter. Both are right. I prefer follicle. So you call it a lymphoid follicle. That seems to be the dominant terminology. In the submucosa is where the location is. From here to here is submucosa, and that is where the nodule or the follicle is located, right? But it does present lymphocytes right up to. This bit is the glandular part, which you are seeing. PV is blood vessels. This will be the rest of the submucosa, and the muscle will start beyond that. So when you get a site for identification, this is the part we will keep, where you will definitely see the huge lymphoid follicle compared to the intestinal gland. But both together is equal to appendix. You need to be able to see both, be able to identify. Otherwise, this could be any other lymphoid organ, right? If this has stratified squamous epithelium, it could be tonsil. Unless you see the gland, you don't know that it is part of it. Get it as a discussion slide. Get all four layers, right? Get the whole thing. Then inside of this, you'll see circular muscle outside. Circular muscle, and outside of that, you'll see the longitudinal. That confirms that this is gut, and therefore you will be able to discuss even those. When you're showing it to an examiner for identification, and when you're drawing it, theory answer, or in your board book, please focus on the intestinal, the luminal surface and the features which identify as part of the. So, finished a little early today. This is what the two parts of your large intestine histology will look like. This is the rest of the colon, mostly ascending transverse, descending and sigmoid pieces. We're not looking at the rectum much. The rectum will also have similar look, but it will have fewer glands and it will have much more of adipose tissue over the submucosa, and it will have lesser lymphoid material, and it will have the same goblet cell kind of a structure. In fact, much more goblet cells, because nothing further is going to be absorbed from rectum, and it's not even water. And these arrows are the openings of each of the intestinal glands. We are not giving them a name like crypt given this. So here we can just call them openings of the intestine. Why opening? Because it is from here that the mucin will be secreted out, and also any local neurohormonal molecules if required. Basically, it is to lubricate the fecal mass. The surface cells, which are mostly columnar, are absorbing water, only water and minerals from the contents of it. No other digested material is capable of being absorbed by colonic mucus. Please remember, all the other components, carbohydrate, lipid, and protein, must finish getting absorbed by the end of the ileum. Colon has no possibility capacity to absorb polypeptides or lipids, micelles, fatty acids, or sugars, right? So only the water part will be absorbed. A is adipose. I think this I've turned that previous slide, turned it that you see from lumen to outside. Show you what the surface mucosa is expected to look like, and the fact that lymphoid follicles very close actually occupy the submucosa, presence of the lining of the rectum and the lining of the anus. We show it to you. Anus, because it is the last part of the gut and it is going to open at the anal. The anal canal, which is proximal after the, has a very rich venous plexus just under its submucosa. Has a lot of venous there, which is very important in a disease called piles. But we don't have a slide to that. Just remember that it has a rich venous plexus, which is an interface between gastrointestinal venous drainage and body surface venous, stomach and visceral venous territories come connect at the anus. They also connect at the esophagus, at the upper end of the gut. Those are two areas which we can expect to see. Logically, we may not be able to see it so significantly, but it shows up in disease. In the intestine, the disease which happens commonly, not so commonly, but often enough, is called ulcerative colitis, autoimmune phenomenon, where the mucosa itself being attacked by the defense system, by the WBC, and forming ulcers in the mucosal lining of the large, and that's why it's a colitis, inflammation of the. And you will look at that in pathology. That is one disease. Plus, there are other diseases of the colon which may reflect in history. Sometimes there are incoordination, discordance between the peristalsis of the food, which gives rise to different types of intestinal obstruction in the large intestine. So because of a mismatch or a misfiring between the nervous plexus, but that doesn't show up in history. That happens in certain congenital, happens as a consequence of worry about that. Obviously, in all this histology, you did not see any bacteria, but in life, there was a whole bunch of bacteria occupying the colon. By the time we process sample tissue, we wash out everything which was in the lumen of it. Those bacteria, you must remember, are on the surface. They are never inside our own tissue. And those surface bacteria are the ones which we don't see them. We see colonies of bacteria or pus pockets, then that's a pathology. It is not healthy histology. In the appendix, also we know, I think many of us, some of us, have had appendicitis once in our life. That's when the incoming infection has overwhelmed the defense available and has inflamed the lining of, and those inflammatory changes are pathological. We don't usually do a biopsy to usually deliver the whole appendix and study it later, and that will show an appendicitis. But don't worry about it now. Just remember the normal healthy histology, the colon of the appendix. You have to decide and interpret. Please look at your histology atlases. Do the drawings, label them properly. If there are any points I missed or anything, send it to me in your chat and we'll take up any. So far, I have not received any doubts from it. So either everybody sleeping off or just logging in their attendance and then running off. Don't do that. These things won't be repeated. It'll be very hard for you to understand them all. So please attend these classes and do a little bit of minutes per lesson to help you to anchor it. Come back to college, you're ready to. I also wanted to wish you and your families, those who are celebrating, happy. I'll see you on Wednesday. And I wish all of you are, I hope, and your families are healthy. Nobody has got corona. Have a happy week. I think there's another class in the afternoon. Please attend that also at 2 o'clock.